The overall goal of the following experiment is to map the distribution of DNA repair proteins around double-stranded DNA breaks at the nanoscale in order to study the ultrastructure of damaged chromatin undergoing repair using electron spectroscopic imaging. This is achieved by generating DNA expression constructs coding for fusion proteins of DNA damage response proteins using a fluorescent protein and the MiniSOG tag. As a second step, constructs are introduced into cells by transfection, and then the cells are damaged by either laser or gamma irradiation. Next, the irradiated and fixed cells undergo electron microscopy preparation steps in order to make the distribution of the MiniSOG tagged repair protein visible in the electron microscope. The resulting electron spectroscopic imaging provides detailed images that allow for the analysis of the chromatin ultrastructure and the relationship of specific proteins with DNA repair foci.
The main advantage of this technology over other existing technologies like immunogold labeling is that it's not dependent on the accessibility of antibodies and that it labels the structures of interest quite densely. Generally, individuals that are new to this method will struggle because the photoexcitation is very sensitive to pH, light intensity, and oxygen saturation.
Visual demonstration of this method is critical because the photooxidation, oxygen saturation, and illumination steps are sensitive to very small changes. And so it's important to illustrate how the method is done in order to reproduce it. Dr. Hilmar Strickfaden, a postdoc in my lab, will demonstrate.